Hi, welcome to my poster. My name is Peng Han, and I would like to share my research with you. Recently, there are many methods proposed to integrate single-cell multiomic data. However, how to incorporate prior knowledge and decompose the variability explained by different modalities are still an open question in the field. Here, we propose a novel probabilistic learning framework that creates graphical model explicitly based on the prior knowledge of their data type. The prior knowledge is provided as a DAG or a directed acyclic graph, where each vertex represents a data modality and a directed unweighted edge represent the potential influence between the two modalities. For example, here we hypothesize that the chromatin accessibility data may influence the regulation of gene expression in general. Our framework will create a hierarchical variational autoencoder according to our hypothesis to learn the conditional generative model of the observed data. The model is trained by optimizing the evidence lower bound with the sequential training based on the topological sorting order of the modality. Take our previous input as examples. We first train the neural network layer of the chromatin accessibility data and freeze the RNA gene expression data. Then we sequentially train the gene expression data and froze the chromatin accessibility data. We apply our method to the SNR-seq dataset and show that our method can decompose the joint latent distribution here on the left to latent representation of the chromatin accessibility and gene expression on the right, the two right panel. Note that the latent representation of the gene expression is aiming to learn the residual of explained variability given the posterior of the chromatin accessibility data. This implies that the distinguish of these cells from other cell type is not only caused by changes in the states of chromatin accessibility, but involves other regulatory mechanisms. For more application of our methods, please refer to our LMRL workshop paper or send me an email. Looking forward to have more discussion with you. Thank you.